Good morning. Welcome back. Once again, full disclosure, empty room, locked door. I'm by myself. Chapter 3. Hang on just a second. Let me adjust this camera. All righty. So let's talk about the cell. Food for thought. In our intestines, there are bacteria that make vitamins and keep us healthy. There are other bacteria that make toxins that can contaminate foods. So we have to keep a healthy balance in our intestines of bacteria. An example, each year about 265,000 people in the United States become affected with a toxin-producing E. coli. And we will talk about cell structures and the components of a cell as we move along. So the cell theory. Cell theory states that each organism consists of cells. The cell is the structural and functional unit of all organisms, and in other words, all organisms either are a cell or are made up of cells. All living cells arise by the division of pre-existing cells, and cells contain the hereditary material needed to produce a new line of the same organism. All cells can have several components in common, and then there are some cells that have unique, and I will tell you which ones are unique and which ones are common as we move along. The plasma membrane surrounds the cell, maintains the boundary of the cell. It has a quality called selective permeability, which means that the cell controls what comes in and out. Proteins embedded within a lipid bilayer are attached to one of its surfaces, carry out many membrane functions. So within that plasma membrane, there are proteins that have specific jobs, and we'll point those out in a minute. Inside the cell, there is a jelly-like mixture of water and all sugars and all sorts of other dissolved products. That's called the cytoplasm. It's the site of some or all of the cell's metabolism, depending again on the type of cell. Prokaryotic cells that don't have any membrane-bound organelles and do not have a nucleus, it's in the cytoplasm where everything takes place. In yours and my cells, we have organelles for generalized function, but there are still some things that do happen within the cytoplasm. An organelle is a specific structure, and we'll talk about some different organelles in a minute, that carry out a specialized metabolic function within the cell. Eukaryotic cells have a nucleus that contains the DNA. Again, the difference between a prokaryotic cell and a eukaryotic cell. Prokaryotes, no nucleus. Eukaryotes do in fact have a nucleus. There is a picture of a typical cell. This is actually a plant cell and shows the plasma membrane. That's going to be this membrane right here. This sort of thicker area out here is the cell wall. Not all cells have a cell wall. This is basically showing you the components common to all eukaryotic cells. All eukaryotic cells have a cytoplasm, they all have a DNA, and they all have plasma membranes. Some of these other things we will talk about in just a few minutes. So, cells must be small to efficiently exchange materials with their environment. Cells have a prescribed surface area to volume ratio. That's the relationship between the surface area and how much it's holding. The relationship, again, when we talk about cells, and a little bit later in the semester, we will talk about how the shape and that surface to volume ratio plays a role in how the cells can do their jobs. But this surface to volume ratio limits the size and influence of cell shape. So some cells are able to expand greatly. Other cells can expand, but not so much. Some can't expand at all, and it all has to do with that surface area to volume ratio. Again, constraints of cell size. You can see there's 3 different cells, one with a diameter of 2, I don't know the centimeters, 1, 3, and 1, 6. You see the surface area increases, but the volume, again, you have a surface area to volume ratio here that looks like it's about 3 to 1 or 3 to 2. Here you've got a 2 to 1 ratio, and here you've got a 1 to 1. Oh, it says right here it's 3 to 1. And 2 to 1 and 1 to 1. So again, this is going to constrain as you get closer and closer to this 1 to 1 ratio. That's going to be ultimately how big a cell can actually be. So no one knew cells existed until the microscope was invented. There was no concept of cellular life until the late to mid 1400s. We see cells using multiple different types of microscopes. If you want to see the molecules of life, such as lipids and carbohydrates and proteins, and you want to see viruses, you have to use what is called an electron microscope. I worked with an electron microscope when I was over at UALR. We do not have one on this campus, nor will I suspect we ever have one at UAPTC. They cost in the range of $2.5 to $3 million just to purchase, and then they cost several hundred thousands of dollars every year to maintain. UALR, I believe, still has one. I'm not sure that it's been properly maintained over the years. Again, costs a lot. I know UAMS has one or several. But again, can't see an atom. This is an atom, and you can't actually, that's a molecule of water, and you can't see that because it goes beyond the range of the electron microscope. Then if you want to see bacteria or some organelles, you can see some organelles and bacteria and most eukaryotic cells you can see with the light microscope. Things that can be seen with the human eye, here's a tree, that's probably a blue whale, excuse me, a giraffe, a human, a duck, a mouse, a frog, an ant, that's a frog egg, and that's some sort of an insect. It looks like some kind of a louse, I suspect. Well, yeah, it's Edward Louse is the one who made this slide. So the cell membrane, it is a mosaic of proteins and lipids, mainly phospholipids. It functions to selectively control what comes into and out of the cell. We call that selective permeability. It is referred to as a fluid mosaic model. If you've ever taken an art class, you know that a mosaic is a combination of things. You put a bunch of different shapes together to make a picture. That is a mosaic. And that's what we're having. You've got the phospholipids, you've got proteins, you've got cholesterol, you've got lots of different things that make up this cell membrane. The reason it's referred to as a fluid model is because the individual components are not anchored together like bricks in a brick wall that are mortared together. These things are simply held together by pressures. And there are times when they may open just a little bit and then close. Again, constrained by that surface area to volume ratio. But these things are not anchored, they're not tied to each other. They're just simply held together by pressures. There again is a picture, we've seen this before, of the phospholipid bilayer that makes up the typical cell membrane structure. Again, hydrophilic end, hydrophobic end, hydrophobic end, hydrophilic end. There's water inside the cell in the cytoplasm. Most cells are surrounded by water. Most, not all, are surrounded by what is referred to as an extracellular fluid, and that extracellular fluid can in fact, or does in fact contain water. Then here we show some of the other structures, some of the protein structures that, again, here's the phospholipid bilayer. You have the different protein structures. This is some sort of recognition protein, such as the A or B antigens that are found on the surface of red blood cells, for instance. This is what is referred to as a transport protein. You see there's like a little channel in it that allows large things to get into and out of the cell. And then there are some other types of protein structures as well. Prokaryotes found in domain bacteria and domain archaea. These are the smallest and most metabolically diverse of the cells. They are found virtually everywhere. You go up into the Arctic or the Antarctic and you drill ice cores and you melt the ice, you'll find bacteria living in those ice cores. Now, they're not actively metabolizing while they're frozen, but you thaw them out and they start to metabolize very quickly. They basically reanimate. Many of the archaeans are what are called extremophiles. They live in those very extreme environments, such as hot springs or an area where the pH is really, really high or the salt content is really, really high. There's a picture of just several different types of prokaryotes and their vast array of sizes and shapes. We're not going to worry about what these things are, but this one is a wonderful example. If you look very closely, you see a very long structure that's called a flagella. And then you'll see numerous shorter structures, which in the prokaryote world are called pili. In the eukaryotic world, we refer to a structure like that as a cilia, and we'll talk about cilia in a few minutes. So within the cell, you have the cytoplasm. There are some ribosomes. We need to be real careful with the word ribosomes because, yes, they are found in prokaryotic cells, and yes, we've been told that there are no organelles in a prokaryotic cell. And then in just a few minutes, I'm going to tell you that one of the organelles found in eukaryotic cells is something called a ribosome. So let's be real careful with that word. Yes, there are ribosomes found in the cytoplasm of the prokaryotic cell. And yes, we do call it an organelle of protein synthesis, but it's not technically an organelle. We're not going to go into the great detail needed for that difference. Just believe me when I say that they are a better word there would be the site, S-I-T-E, of protein synthesis rather than the organelle of protein synthesis. All prokaryotic cells have a circular DNA. Their DNA kind of looks like a rubber band. You take the rubber band off the newspaper. Yeah, no, not many newspapers being delivered anymore. But when you take a newspaper, it's not a perfect circle. It's sort of a round structure. It's kind of flattened. And that's kind of what the DNA of prokaryotes look like. And they may contain additional genes in structures called plasmids, and we'll talk about plasmids a little bit later in the semester. Your prokaryotic cell may contain a cell wall. Not all prokaryotes have those. This is a semi-rigid but permeable structure. It surrounds the plasma membrane, again, of some cells. It consists of peptides and polysaccharides in bacteria and proteins in archaeans. That is one of the difference between the archaeans and the bacteria is what's in the cell wall. When a cell wall is present, what's there? If it's a protein and polysaccharide, it's a bacteria. If it's, if it's a polysaccharide, it's a bacteria. If it's primarily protein, then it is an archaean. Some bacteria have an additional layer called a capsule, which is actually found outside the cell wall. These are structures that tend to help the bacteria stay in place. They're sticky, so they help bacteria form together to form what are called bacterial plaques. We'll talk about those in a little bit later in the semester. And they also may just help an individual bacteria stay where it is. It gets into a hospitable area, an area where it likes, and it will stick to the surface so it doesn't get washed away or moved away. Surface extensions, we talked about, I showed you the pili a minute ago on the bacteria. These are protein filaments, help the cells cling to or move across surfaces, or for the plasmid to transfer. We'll talk again about plasmid transfer later in the semester. Another surface extension would be the long, slender cellular structure known as a flagella. We'll talk about flagella as we get on into the eukaryote cells as well, but basically a flagella is for motility. One of the things we test for in bacteriology, for instance, is we want to determine if a patient has a bacterial infection or there are bacteria present somewhere, let's put it this way. Are those bacteria infectious to human beings? Well, one of the tests we do is to determine whether or not the bacteria have a flagella. because most of the bacteria that are human pathogens are flagellated. They have a flagellum. So if you have bacteria, contaminating a water source, for instance, and you test those bacteria and you find out, well, yeah, there's a lot of bacteria in that water, 99.99% of them are not flagellated, then that water, although I'm not going to drink it, might be safe for human consumption because, yes, it's got a lot of bacteria in it, but those bacteria are not human pathogens. There's a nice little picture of a typical prokaryote showing the cytoplasm with ribosomes involved. There's the DNA. You see it's they've got it drawn sort of a circular kind of shape. You see the plasma membrane, which is the innermost layer, and then the cell wall, the capsule. You see the multiple pili growing from alongside, and then the one very long flagellum. A biofilm is something that occurs, it grows in different environments where not only multiple bacteria, but multiple types of bacteria will grow together and form a layer on some sort of a solid surface. It will provide advantages to the community of bacteria. This one may be producing in excess of something that this one needs, and this one's making in excess of something that this one needs, and this one is making in excess of something that this one needs. So you wind up with a community living together. A wonderful example of a biofilm is when you wake up first thing in the morning and you're yawning and you haven't quite got out of bed yet and you rub your tongue over your teeth and you feel that sort of rough, yucky feeling on your teeth. That's a biofilm. If you've ever walked across the stream and the stream has little pebbles on it and the pebbles feel kind of slippery to your feet, again, that's a biofilm more than likely. Eukaryote body plan. This is a cross-section of a typical eukaryote cell showing multiple different types of structures. Number 1 is the nucleus. Number 3 looks like a rough endoplasmic reticulum. Number 2 is some sort of a vacuole. Maybe we'll get a further, vesicles. There's descriptions now. Vesicles forming. Number 3 is the ribosome. Okay, it's the ribosome in the endoplasmic reticulum. Number 4, that's the endoplasmic reticulum. That's actually the smooth endoplasmic reticulum. The rough one is the one that has all the ribosomes embedded in it. Number 5 is a Golgi apparatus. We'll talk about what that is in just a second. And then number 6 is the mitochondria. The nucleus. Within a nucleus, you will find pores and receptors and transport proteins, which help things move into and out of the cell. DNA will produce something called RNA. That RNA will exit through a pore, where it will then meet up with other products to make, or other organelles to make the proteins that the cell needs. The nuclear envelope is a double membrane. It constitutes the outer boundary of the nucleus. There is a photomicrograph of a typical nucleus of some eukaryotic cell. You see the nuclear pore. You can't see it very well, but there is an opening right there that's going to allow things to move out of the cell. There's the DNA and then the nuclear envelope. The DNA in this particular cell is in a very relaxed state. As we move along in this chapter, we're going to talk about what happens when the DNA condenses and you can see it more readily. The endomembrane system, these are the membranes that form different organelles, includes the rough and smooth endoplasmic reticulum, vesicles, and Golgi bodies. The endomembranes are interacting organelles which make and modify lipids and proteins. They recycle molecules and particles such as worn out cell parts. They also, cells undergo something called endocytosis, where they bring things into the cell. If something gets brought into the cell that the cell doesn't want, there is a process of getting rid of those through this endomembrane system. And it also plays a role in inactivating certain toxins. The endoplasmic reticulum is a system of sacs and tubes found in the cytoplasm of the cell. The rough endoplasmic reticulum is responsible for the production or the making of proteins. The smooth endoplasmic reticulum is responsible for making lipids and fats, and it does not have any ribosomes. The item that makes the rough endoplasmic reticulum have that rough, pebbly appearance are ribosomes. A vesicle is a small membrane-bound sac-like organelle that stores, transports, and degrades things. A vacuole is a fluid-filled organelle that isolates or disposes of waste materials. So vesicles store things. A vacuole gets rid of things. And a lysosome, is a specialized vacuole found in animal cells primarily that contains enzymes which help the internal digestion of a product. Something brought into the cell that the cell doesn't really want, a vesicle will engulf that, bring it inside, and digest it and eliminate it. But again, lysosomes are found primarily in animal cells. A perioxisome is a vessel that is containing amino acids. These play a role in breaking down things like amino acids, it's an enzyme-filled structure that breaks down amino acids, fatty acids, and toxic substances. This is the component that we have that helps us digest some of the more complex things in our diets. The Golgi body is an organelle that modifies structures. We're going to talk about protein production at some point, and we'll talk about, we talked about that long chain protein. That's primary structure, and then it starts getting twisted into a secondary structure. The secondary structure will get turned into a tertiary structure, and the tertiary structure gets modified further to become the quaternary structure. Well, all of that secondary to tertiary to quaternary development all happens at the Golgi body. The mitochondria and the chloroplasts resemble bacteria. There are some theories that they may have actually at one time in the very, 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 very distant past actually been free-living bacteria that were brought into a cell, were not consumed or digested by the cell, and the cell and the bacteria formed a union called a symbiosis, and that symbiosis has just come forward in evolutionary history. The mitochondrion is a double membrane organelle that produces ATP. It is the site of energy production in our cells. Think of it as the battery or the powerhouse, the power supply for the cell. Chloroplast is an organelle of photosynthesis. You and I do not have chloroplasts in our cells. Plant cells do, and lots of protist organisms do. But again, photosynthesis occurs in the chloroplast. Again, cross-section of the mitochondrion showing the outer membrane here in the sort of blue layer. Then you have the outer compartment. The outer compartment is going to be the area between the outer membrane and the inner membrane. So this sort of fluid-filled area is going to be the outer compartment. Then you have the inner membrane, and within the inner membrane you have the inner compartment. There is a cross-section of a chloroplast, again showing the two outer membranes. The stroma is going to be the fluid within these. Then the inner membranes are going to be all these membranes that make up these little structures that kind of look like pancakes, or stacks of pancakes, or stacks of coins. Cytoskeleton is a system of protein filaments that supports, organizes, and moves eukaryotic cells. Some eukaryotic cells have the ability to move from place to place, and the way they do that is through the interaction of maybe they have a flagella. They also have the ability within their cytoskeleton to make the cells expand and then contract, or will make one side of the cell expand and then the other side will follow it, sort of like an ooze kind of effect. The cytoskeleton has several different components that interact. We have these things called motor proteins, again, that play a role in movement. You have transport protein that move items from place to place rather than the cell itself. Microtubules are hollow filaments of tubulin subunits. All I want you to remember about microtubules is that they are hollow. Microfilaments are reinforcing skeletal elements, and intermediate filaments lock cells together to form the tissues that we talked about. You know, you have a bunch of cells form a tissue. Well, the way those cells hook together is through their intermediate filaments. Microfilaments, again, are reinforcers, and microtubules are hollow. There's just again, picture of a fluorescent micrograph, microtubules and microfilaments. The microfilaments are in blue, microtubules are in yellow. These different elements support and guide the cell's length in a particular direction. So again, you see the cell is sort of moving this way. It will get to a point where this end will anchor and everything back here will follow in behind it. Motor proteins are proteins that interact with the cytoskeletal elements to move cell parts to cell whole. That shown for you there is a motor protein that moves a vesicle along a microtubule. Basically what will happen, this thing will flip and then drag and flip and drag like that. Cilia are short hair-like structures that project from the plasma membrane. They will coordinate. They will set up a sort of wavy fashion like this, and they basically create a current which will do one of two things. The current will either move the cell itself from place to place, or will move the cell. Our cilia, the cilia in our system, aren't designed for moving cells, they're designed for moving fluids. So the cilia will create a current which makes things move past the cell. Flagella is a whip-like structure that does like this. It whips back and forth, and it creates a swimming motion. That just happens to be a human sperm or a sperm cell, and I suspect it is a human cell. Pseudopods are temporary projections. All of these little things like here, those are all pseudopods. They form temporarily. They'll do one of two things. Again, if the cell is sending out one like this, it's probably going to move. This will move up here, anchor itself, draw the rest of the cell with it. The rest of these can engulf prey items. These two, for instance, could come together. You can see where there's a little line, looks like they're fixing to start forming a projection here. They will come together, and whatever's inside this little pocket right here that's made will then be brought into the cell and used as food or nutrition. The extracellular matrix is again, supports cells and tissues. It functions in cell signaling. If you've ever heard of tissue rejection when somebody gets a kidney transplant, for instance, it is the extracellular matrix, or the ECM, of the donor tissue is not being recognized by the host tissue, and they're doing that. They're fighting that battle. The cell wall, again, not found in you and me. It's found in plants and fungus organisms, and some of the protists have a cell wall. It protects and supports and gives the cell its characteristic shape. For instance, if you were to look at a cross-section of a potato, you would see that all the cells have a little hexagonal shape, sort of a six-sided shape to them. It's not a perfect hexagon, but they are sort of hexagonal in shape, and it's the cell wall that gives them that shape. There's just a drawing of the extracellular matrix. You see the cuticle on the outside. This happens to be a leaf. And you have the outer membrane, and then you have the photosynthetic cell within these things called palisade cells. But again, you'd have another layer of these type cells here on the bottom if the slide picture extended far enough. Cell junctions. There's several different types of ways cells anchor themselves together. There's something called a tight junction. A tight junction is a set of proteins. It joins epithelial cells, prevents fluid from leaking. The junctions that are holding our skin cells together are tight junctions. An adhering junction is a cell anchors cells to each other or to an extracellular matrix. And then there is something called a gap junction, which is a channel across membranes of adjoining cells. And gap junctions generally is a way of passing material from one cell to another. It's kind of like a channel way. So you've got gap junctions, adhering junctions, and tight junctions. Again, there's just an example of the different types of junctions. You see the tight junctions there at the top, the gaps. If you see, there's a nice little hole in that can open and close. And then you have the adhering junctions. These are just anchoring the two cells together. There's no communication going on between here like there is there. In plants, there's a structure called the plasmodemata that connect the cytoplasm of adjoining cells. The plasmodemata, think of it as an open channel. It is a structure that is open from one cell to the next. It's always there. Allows primarily for water, but other things to move from one cell to the next, and then on to the next, and on to the next. There are 6 properties that characterize living things as different from non-living. Again, this is that cellular structure. Make and use organic molecules. We have to be able to make certain complex molecules, and then we have to be able to use them once they're made. Life consists of one or more cells. There must be some self-sustaining biological process like metabolism. You know, we have to have a process for using things, getting rid of the waste materials. That would be metabolism. All life must change over the life of the organism. Again, develop, mature, and age. We have to use DNA as the hereditary material for reproduction. Again, that's where viruses fail to meet the categories of life. And they have a collective capacity to change over successive generations. In other words, cells can modify, evolve, or mutate. A couple of things to ponder as we go on from one chapter to the next and one day to the next. Plants contain a very large central vacuole. What is the function of this organelle? Remember, a vacuole is a storage product. And the central vacuole of plants stores primarily water. So what is the function of this organelle, and why do plants need one but not animals? Well, plants need one because they're anchored to the ground. And if the ground is not there, they've got no moisture. And so when moisture is plentiful, the plant will pack on lots and lots and lots of different water, hold on lots of extra water, and in doing so, then when the soil dries out, the plant has a supply of water. Animals, on the other hand, don't need that structure because they can move from point A to point B to the water hole. They can go from where they stay every day to a place where there's water, and if that water hole starts to dry up, they can move to another one. They have another source of water. Another point, hand washing helps reduce the risk of bacterial infection. Should be a period right there. The FDA has recently ruled that antibacterial soaps offer no benefit beyond that of normal hand washing. That wasn't all that recent. She'll be 30 this year. My 30-year-old daughter did an experiment in the 5th grade, I think it was, where she showed the efficacy of hand washing when it came to bacteria, and her experiment showed that as long as you were using hot water and soap and vigorous friction as you were washing your hands, the type of soap really didn't matter. It was just as effective using a cheap soap from the Dollar Tree and the most expensive antibacterial soap you could find on the shelf at the store. You got the same result if you used hot water, vigorous friction, and any type of soap. So what effect does soap and hand washing have on bacteria? And why might antibacterial offer no better solution? Hand washing is a process that when you take if you were to take my microbiology course, we would call it degerming. Because hand washing in and of itself does not kill the bacteria. All you are simply doing when you're washing your hands and then rinsing them off is you're just getting the bacteria off your hand. You're not killing them. You're just rinsing them off. For an antibacterial soap to be effective, you have to put it on your hands and then wash your hands, and the bacteria would still be there. They'd just be dead or inactivated. Well, that's not what happens in hand washing. We rinse them away. All right. So that wraps up this chapter on the cell. Again, if you have any questions, email me in either the Blackboard contact area, the message board, or directly on my email. Keep in mind there are some online assignments through your MindTap, I believe is the process, but it's listed as assignments, so make sure you get in those reading assignments and get those few extra points that you may need toward the end of the semester. And have a good day.